Okay. All right. So, can you hear me? Everything okay? You can see the screen?
Yes.
Lovely. Now, just before we get too far into this, I want the names all will more or less come up later, but at the bottom of the screen, you'll see a longitudinal section of the right kidney. And just in case you don't remember or you don't know, when we talk about anatomical features, it's as if you are a person looking at another person's anatomy. So the right kidney is such that these vessels here face in towards the middle. Okay, so if you're looking at a person, their right kidney would look like that. So it might look a bit odd to you, but never, you may not. All right, so the number of pictures. So here we have the right kidney and the left kidney. So it's the opposite to us. And as you see, the vessels come in through that. So the system we have is the kidneys, two kidneys. There are the red one's the artery, that's the aorta, and then the renal arteries, there and there, left and right. And then this keeps going down to the legs. There's the bladder with the ureters coming through and the urethra. The blue one is the main vein that collects all the blood from the legs and the lower part of the body and goes back to the heart. So we have all sorts of wastes in our bodies. One of the main ones is carbon dioxide, which we breathe out. The other major one is what we call nitrogenous wastes. Now, as you know, I think you know, proteins are made up of amino acids, and amino refers to nitrogen. So when we break up proteins, excess amino acids are broken down, and and these amino acids are then stored for future use. But if we have an excess, they get broken down and we end up with ammonia. Now, ammonia is very, very soluble and for us quite toxic. So it's the toxicity of it can't last. So it must be removed. Now, this is in all animals. Now, animals that live in water don't have a problem because they just remove it and it's in the water. If you have a normal cold water tank and have goldfish in it, it doesn't really matter because the goldfish have got a very good capacity to withstand the ammonia in the water, but also you need to have a filter to remove it. But with what are they called? Tropical fish tanks, which have warm water, if you don't have a particularly better filter than the goldfish, you'll kill the fish because the ammonia just accumulates and knocks them out. Now, in mammals, the ammonia is converted to urea, and it's also water soluble because we urinate it out. But too much urea is toxic, so we must get rid of it. Now, animals that live in very arid areas, or we don't have much access to water, so they can't get rid of urea. So they've developed a different one called uric acid, and that's excreted in, as I say, in solid form. So if you've had if you've ever seen, for example, fly droppings, the tiny little black spots. They are uric acid, like that. They're solid. All insects are like that. What am I going to say? I've lost it. Reptiles, you get rid of it in uric acid form. And if you've seen a bird dropping on your car's bonnet or somewhere, you'll see a white splotch, and in the middle of it, you'll see a darker area. Now, the white splotch is uric acid, and it eats into the Duco if you don't get rid of it fairly rapidly. The dark spot is actually the fecal material, the solid material. Now, birds, as in reptiles, only have one opening. As we have two, they have one, and they everything comes out through the one thing. So that's why one opening, and it's also their reproductive opening. So it's very insoluble. Birds, reptiles. So I mentioned insects, arachnids are like the spiders and that sort of creature. They all remove the waste through uric acid. Now we have another nitrogenous waste called creatinine. Well, that's not the waste. Creatinine is a form of ATP, but it's different. Instead of adenosine, it uses creatinine. And in our muscles, the red muscles, the heart muscle, and the brain, we store this for emergency use. So, for example, if you decide to run for 100 meters as fast as you can, or just start running for the bus or the toilet or wherever you're running to, and you've been quite sedentary, but you start running, there isn't enough time for the ATP to move, to get enough energy to run or to move very quickly. So the first 10 seconds or so is creatinine phosphate, which is in all the cells. And off they go of the cells of those three parts of the body. So it allows us to move fairly quickly, fairly rapidly, quickly in time, rapidly in speed, and it gets us out of trouble. So if you're on the savannas somewhere, you're grazing, and a lion happens to be running towards you, you're not going to hang about waiting for the ATP to start kicking in. The creatinine phosphate will take you, and you get far enough away in the first 10 seconds. So if you been watching the Olympics, those guys that run 100 meters, if they didn't warm up, they run the 100 meters, but it's usually around the 10-second mark. They will not be using ATP at all, the creatinine phosphate. That's why they warm up, because the ATP is much better at providing the energy. And they hardly breathe at all during those 100 meters because it's not using the oxygen as well either. Now, eating red meat, the creatinine that's in the meat, as you said, the red meat, it will get rid of it through the kidneys. So we don't actually use it, but we keep some of it. And so when you have certain conditions like diabetes, or if you're dehydrated, there's an enzyme called creatinine kinase, and what it does, what happens is it increases in these conditions. So it's one of the part of the blood testing that's regularly done by doctors. and it's excreted through the urine, so it's there too. Okay. So we have the excretory the whole system is mainly to remove nitrogenous waste, and mainly it's urea. With us, it also controls our water balance, the body's water balance. I've got too much, I've got too little. And also the electrolytes, the minerals in our body. It controls how much we've got. And if there's too little or too much, it either gets rid of it or it gets in some way of stimulating us to eat it. And of course, a very big part of the kidney is controlling blood pressure. So what have we got? We've got kidneys, and we've got the units, then called the nephrons, and there are about 1 million of these nephrons in each kidney. And the kidneys are around the size of your fist. And so you've got a middle of those, 1 million of those. We've got certain blood vessels, we've got ureters, we've got bladders, one bladder, 2 urethras. So 1 urethra, 2 ureters. And in another diagram, I should also mention, here's so you've got your left ureter, left kidney, right kidney. These yellow pyramidal shaped things are the adrenal glands. And you've got the arteries, everything else I've mentioned. There's, if in the female, the uterus, the oviduct or fallopian tube, and the ovaries. And of course, the bladder. Here's an oldish diagram I found some time ago, but it gives you an idea of, so here's a mouse I'm using, that's about the size of the kidney. So let's look at the anatomy of the kidney. It's got 2 basic layers, the cortex and medulla. Now, in many organs that we have these layers. The cortex is the outside layer, which is this layer here, and the medulla is the inside layer, and they each have different functions. So it could be the adrenal gland, where you got the adrenal cortex, adrenal medulla, you've got the pituitary and so on. So within each of these are the 1 million nephrons, and they get collected, and then you go down the ureter to the bladder. This area here is called the pelvis, the renal pelvis, and this is where you see also the blood coming in and out. I don't think I need to tell you any more there. Now, let's get some details. We have the nephrons, and so what happens is everything's supplied by the blood, and some things are removed, some things are added, and it depends on the status of the body at that particular time. So we have urea, as I said, and water, glucose, sodium, potassium, all of those move through this thing called the glomerulus, and from there to the tubules. Now, this fluid, they'll be diagrammed in a second. The glomerulus has filtered this material out of the blood, and because it's been filtered, it's now, the official name is the filtrate. And this filtrate that moves through the tubules, which you'll see, and the glucose, which is the last thing the body wants to remove, is reabsorbed very early on in the piece. And it reabsorbed back into the blood. And then from there, water and sodium are regulated to maintain our blood pressure. More detail. Okay, so here's this diagram again. This is the kidney with all the bits and pieces, but in this here, in the medulla, the cortex medulla, you see the line here. This section's in the cortex. This section's in the medulla. This thing here, the round thing, is the glomerulus, the red thing. I'll show you a better one here. Okay, look at this here. So we have a thing called Bowman's capsule, and the blood moves in and out through that. And this little ball, very tight ball of capillaries is called the glomerulus. Now, first of all, the Bowman capsule is very easy to imagine. This is called an invagination. So imagine if you've got a balloon, like a kid's party balloon, but partly blown up. And you can hold it in your hand, and you can put your fist into it, like that. So when you do that, you actually have the two walls. I mean, same balloon, but with a fist in there, you have the two walls next to each other. And where my fist is, that would be the glomerulus. Okay. Now, the blood comes in. Now, this word afferent and efferent refers to blood in and blood out. So anything going in is an afferent, in this case, arterial, and efferent is coming out. What you see here, and it's not that clear, but the clearest one I could get, the diameter of the efferent arteriole is smaller than the afferent. So the blood coming in here, the blood's coming in here, and it's under pressure because there's less space for it to leave. And that causes a pressure in here, in the glomerulus. And that pressure causes the water to leave and go into this tubule here. And then glucose follows, the salts follow, and so on. Okay, urea, everything follows out of there. Because of this diameter difference, the pressure is pretty high. So everything's moved. So here we have the glomerulus inside the Bowman's capsule. Now these are tubules. So coming through here, I'll go into more detail in a minute about how this works. It comes down here. There's a big loop here called the loop of Henle. Over here, you've got the proximal and the distal tubules. Eventually it comes through here, and it's a collecting duct, and it gets bigger and bigger and bigger because each of these is from a different nephron, and then eventually goes to the renal pelvis, and from there into the bladder. What happens in this area here is what this is all about. Here's another diagram. This is quite exaggerated, but it's very accurate. It shows what happens. This is the afferent, the blood coming in, glomerulus, and the blood going out in the efferent. And you can see the difference in size. Very important. This shows you also what happens in this capsule, Bowman's capsule, which is this invagination here by the glomerulus. So you see the filtration of water, the salts, and the glucose. Now, as it comes through in the kidney, the other kidney tissue, there are pumps, and there are active pumps, and water though is moved through osmosis. So by moving in and out through here, and H, by the way, is hydrogen. That's the acid. So it keeps the balance of the acid right through this whole thing. If you're not sure, NH3 is ammonia. NH3 is ammonia. K plus is potassium. That's water, H2O. Sodium is Na plus. And of course, you get glucose. So the length of the loop of Henle, which was this one here, remember it's thinner, this is here, and previous diagram, very long one. The length of it depends on where the animal lives, not us so much, but we can modify our environment. So the loop of Henle determines how much water is moved out from the filtrate into the blood. So animals that live in the wet areas where water is not a problem, they have quite long loops of Henle to get rid of the water. Just remove it, you don't need it. And they urinate a lot. And the urine itself is quite dilute. On the other hand, animals that live in arid areas where water is a major problem have very short loops of Henle. And because they're so short, they lose very, very little water to remove the urea. So they end up with concentrated urine. Before I go, there's a tiny little marsupial mouse that lives in this, it's called the Spinifex hopping mouse. And it virtually never drinks. It gets all its water from the seeds that it eats, because the seeds have a large amount of starch. When we break down starch, water is given off, and it's called metabolic water. That's through the process of producing ATP. So that's called metabolic water, and they live through metabolic water. Because I've got these mice, and there are many other examples, but these Spinifex hopping mice have so little water, they may only produce 1, maybe 2 drops of urine a day. But they have developed this amazing change in their physiology to be able to survive with very, very high concentrations of urea in their bodies. If we had that sort of concentration, we'd be long dead. We just can't. And so these mice are mammals. I think, I'm not sure. Okay, doesn't matter. So the whole idea of urine production involves osmosis. Now, you've all heard the word osmosis. I don't know how many of you understand what it actually is, but you can't put your notes under your pillow the night before the exam and hope that everything in the notes moves through the pillow into your brain through osmosis, which is an old joke. But osmosis works. You need to have a membrane, and you need to have water, and you need to have some molecules like salt. Well, salt is not a molecule in water because it's ions or glucose. And the concentration difference between the two sides gets movement of water going, and that's osmosis. Okay, and depending at what the difference is, you end up with an osmotic pressure. And the greater the pressure, the more movement there is. So I'll go through osmosis because so few people really have an idea of what it's about. But we operate under this process of osmosis. Our system, the whole body works on this, and it controls water across all our membranes. So the rate and direction of the movement of water, it depends on the difference in the concentration of a solute. Now, we have water, which we call a solvent, and the solute is the other molecule, like sugar or salt, could be other things, but they're the main ones for us. So we have solutes in solvents, and then you get what's called a solution. So you have your cup of tea, coffee, and you put in, if you're a sugar taker, you put in some sugar, give it a stir, you've got a solution of sugar. Okay. So you have the solution is, the solvent is the water, the solute is the sugar, and the solution is the sweet tea. So when there's a difference in the concentrations, the water will flow in the direction of the greater solute concentration. So I'll show you in a second. So the whole idea is to dilute it, to equilibrate the two sides of the membrane. So when the concentration inside or outside the cell are the same, then the water, it flows all the time, even if it's exactly the same concentration. There's constant movement of water, but the amount of water in and the amount of out is the same. So there's an equal amount of water flowing. Now, in the case of equal concentrations of the solute on both sides of a membrane, it's called, each side is called an isotonic solution, iso equal, isotonic solution. Now, if one side is lower than the other side, I've got diagrams, but I thought I'd mention this first. So one side lower than the other side, that's the concentration of the solute, like the sugar, then the water will flow in towards the higher concentration. So the outside of it is then called a hypotonic. So it's a lower concentration than what's inside the cell. So inside the cell, you get more sugar or salt, whatever. And then outside, so if you just got straight water, tap water or distilled water, then there's this, the water is said to be hypotonic. It's lower concentration. If it's higher inside the cell, or whatever we're looking at, the water will flow out of the cell. So the water outside the cell, or the solution outside the cell, is known as hypertonic. It's higher. So the water from inside flows out. If one day you're cleaning out your fridge and you find this poor old carrot that you forgot about, and you pick it up and it feels like it's like a soft rubbery thing. Now everything in the carrot is still there, it's still alive, just, but it's missing the water. If you take that carrot and put it into a bowl of water, tap water, the water, which will be hypotonic, will flow into the cells of this poor old carrot and fill up the cells continuously till it's all done and it's nice and firm, and you can take a bite of the carrot or use it for cooking and it's as good as new. So if you take a carrot or an apple or something, and you get this crunchy sound, that means the cells are full of water and they're very firm. So here's an example. The isotonic solution is when inside and outside the cell have the same amount of solute, the same concentration. So the water just flows in and out. If there is if there is less solute outside and the water flows in to dilute the inside concentration, then we have, it's a dilute solution, hypertonic, and that will then, if we put a red blood cell, which is a standard way of showing kids at school, or used to be, because they stopped us being able to prick kids' fingers, then the cell will just burst. On the other hand, if you put it into a more concentrated solution, a salt solution or sugar solution, and then which is greater than what's inside the cell, the water will flow out of the cell and shrivel up. And it ends up a red blood cell starts looking a bit like a crown, and that's called crenulation. And down bottom here, you'll see it's been crenated, not with an N, not an M. Very different. So, okay, that's so the solution is hypertonic. All right, so there's a short video here. I hope this works. Okay. Come on. Aha.
Grammarly is by your side, especially when you have a mountain of work and you're short on time. From spelling and
The kidney is the body's filtration system and produces urine in its thousands of nephrons.
It's not coming through, Ruben.
Share that screen, Ruben.
Well, I did click the.
So you need to go and share that screen.
I did click the sound. It says you are sharing sound from the computer. Yeah, you should hear it.
But the actual video is not coming through.
Okay. It's been a while since I've shown a video. Share screen. Where is it? There we go. No, it's not. Okay, can you see it now?
Yes, good.
I'll go back.
The kidney is the body's filtration system
Okay.
and produces
Yes,
urine in its thousands of nephrons, the kidney's basic unit. Each nephron begins with a Bowman's capsule, wrapped around a capillary ball, the glomerulus, which becomes a tubule that eventually forms the loop of Henle, before collecting into tubules and ducts that empty into the bladder. Fluid from the blood enters the nephron by filtration and passes along it, where substances are added from the blood by secretion or removed from it by reabsorption. Potassium and hydrogen ions, organic acids and bases, metabolites and foreign substances are secreted across a selective membrane. The potassium and hydrogen are exchanged for sodium along the tubule. 99% of the filtrate is reabsorbed through a mixture of passive diffusion down an osmotic or concentration gradient and active transport by pumping against a concentration gradient. The kidneys can conserve water. The only way that the body can move water is by moving sodium chloride first. You have to create a concentration gradient so that water can move by osmosis in the direction of the highest concentration. The loop of Henle creates a hyperosmotic region in the medulla of the kidney, acting as a sponge, drawing water out from the collecting duct, if the duct becomes permeable under the influence of vasopressin, also known as antidiuretic hormone or ADH. The ascending limb of the loop of Henle, which is impermeable to water, pumps out sodium chloride via sodium-potassium chloride cotransporters from the urine into the blood in the vasa recta. Blood in the vasa recta carries this sodium chloride into the medulla, making the entire region hyperosmotic. The blood, which is moving in the opposite direction to the urine in the loop of Henle, then ascends parallel to the descending limb of the loop of Henle. This descending limb is water permeable, allowing water to move out of it into the vasa recta. This entry of water into the ascending vasa recta reduces the osmotic potential of the blood to normal as it returns to the body. The interplay between the loop of Henle and the vasa recta is called the countercurrent multiplier. Essentially, the medulla acts as a sponge, retaining and releasing water to address the water requirements of the body. ADH acts on the collecting duct to make it permeable to water, allowing water to move into the hyperosmotic medulla, where it is drawn up by the vasa recta and returned to the body.
With Nordic Track, the results you want start here.
Let's go on a journey through Antarctica.
In Thailand. Okay, so we go back now to all right. You've got the link in the notes, so you might have to watch it a couple of times more just to get the idea. But it's basically the idea that water moves and it's the salts move and the water follows, depending on what the situation is. And I'll get to the hormones a bit later. Now, the bladder is a very useful thing to have, and because it stores the urine for you. It's sort of a little bag. Water comes in, urine comes in through the ureters, and it's stored and then leaves via the urethra. Now there's a sphincter here. Now sphincter is a smooth muscle ring, so it's always closed until you are ready to release it. And there's also another sphincter here, which is in the males under the prostate gland. It's there, and you've got a little bit of extra control. The females have that without the prostate gland, but so you've actually got a couple of controls there. Now, the ureters, sorry, the ureters which come here, notice they come here at near the bottom there. So it's got three openings, two for the ureters from the kidneys and one urethra out. As some of us men get older, the prostate can sometimes enlarge. Now, when it enlarges, it also presses on the urethra, and it makes urination somewhat difficult. There are tablets which reduce the prostate, and that alleviates the situation. And of course, if you have prostate cancer, then the whole thing could be removed or radioactively fixed up, maybe, depends. But that's not our topic today. Now, how much water is involved? What sort of volumes have we got? Now, in an average 70 kilogram man, so an average man is said to be about 70 kilos. So we have about 5 liters of blood, 5 liters. Now, the glomerular filtrate, that means the amount of filtrate, that means the water and the sugars and the salts that going through the glomerular, through the two kidneys, is about 180 liters a day. A very, very efficient filter. Now, if you divide 5 into 180, it'll give you the number of body blood volumes that go through the kidneys every day. An amount of urine that we produce is about a litre and a half, assuming we have enough water, we're not dehydrated, we're not drinking too much. So from the 180 liters, 1.5 liters are remaining, which means the rest has been reabsorbed or resorption. We also lose water through insensible perspiration. Now, perspiration is the sweats. When you're working hard or it's a warm room and you start sweating, you take your jumper off or something. Insensible perspiration is water just leaking out, if you like, through the skin. Your skin's losing water all the time, constantly. So it's not sweat. It doesn't come through the sweat glands. It comes through the skin, and we lose 0.5 liter, maybe a bit more a day. In feces, depending on if it's normal sort of feces, about 100 ml a day. So if you look at that, so if you're losing 1.5 liters there, maybe 0.5 liter here, that's around 2 something liters a day of water you lose. So you've got to take at least that amount in. And that's where we're talking about taking 6 or 8 glasses of water a day. You don't need it because everything you eat has got water in it. So if you have an orange, whatever the size of the orange is water with a solid bit. So that 8 glasses a day of water is long been debunked. You don't need it because you need the water, but it depends on what you eat. All right, so how do we maintain constancy of every part of the body? So the right amount of water in the body, the right temperature, the blood pressure, the electrolytes, the pH, everything is controlled, everything. So the water output, as said, I gave sort of under ideal conditions, 1.5 liters and so on, or 2.2. This one, so that sort of range a day, and what's affected by the volume of your fluid intake and diuretics that you might have. So if you're a coffee drinker like me, we get rid of extra water, which needs to be made up because of caffeine is a diuretic. It causes extra uptake of water into out of the kidney, and alcohol does the same thing. Now, blood pressure is actually controlled by the body, by the kidney in the body. So we have this hormone called vasopressin. It's produced in the brain, and it's also known as antidiuretic hormone, ADH. Now, a diuretic is this, the caffeine or the alcohol. Antidiuretic does the opposite. It stops the loss of water. It increases the reabsorption of water in the blood. So we have antidiuresis. Diuresis is the removal of water. Antidiuresis is the opposite. The other thing it does, it constricts blood vessels. So if blood's moving through your body, and blood moving through your blood vessels, and if it's a particular diameter, that's it. But if your blood pressure drops for some reason, then the vessels, the arteries in particular, are constricted, so the pressure goes up. And that's very important. So this increased blood pressure. We have a system called the renin-angiotensin-aldosterone system. I don't expect you to remember that, but we have a system that's made up of these hormones. And I'll go through it. It's just fascinating how these systems have developed in animals the time that we've been developing. Now, when the blood volume is low, now it means you might be dehydrated, or you might have an accident and you lose blood. But whatever the cause, when the blood volume is low, and that's sensed by the kidneys, they secrete this enzyme, renin. And what the renin does, it stimulates the liver to produce a hormone called angiotensin. And this angiotensin, it goes around and constricts the arteries. And by the constriction, it increases the blood pressure so that the heart can work properly, and the brain also. And it's part of this system which the medical fraternity targets to lower blood pressure. So, where are we? Okay, so the angiotensin, what it also does is stimulates another hormone. It's a steroid. That means it's made from fats, called aldosterone. And this is from the adrenal cortex. Now, remember I mentioned cortex and medulla. Now, the adrenal gland, and I showed you where they are, on top of the kidneys. The outside part is the cortex. So this hormone is made in the adrenal cortex. What aldosterone does, hey, I've got a loose arrow here. So the main role is to regulate salt and water in the body. Okay, so it affects blood pressure, and increasing this blood volume, which is what it does, increases the pressure, not the constriction of the blood vessels, but the amount of blood. That means the amount of water that's in them. And also it reduces the amount of urine that's produced, again, holding back the water. So increase the blood pressure. So it depends on the situation, of course. So let's go through this. Hypo means low volume, so low blood pressure. If that's the case, renin is produced, and angiotensin, the two, that's an enzyme, that's an hormone, and it converts it to a thing called angiotensin 1. And that's converted to angiotensin 2. And what it does in the body, the kidney retains sodium ions via this aldosterone. Remember, this is for low blood pressure. You need to increase the volume of water in the body, in the blood. And then there's so increase retention of the sodium. And then there's water retention, because the sodium draws the water. Remember, we talked about hypertonic solution. And then increases your appetite for salt. You need to have more salt. You feel like eating something salty. And then, having done that, you feel like increasing your water intake. And that, all that together, will increase your blood pressure. Okay, that's all really nice and good. But what happens if your blood pressure is too high? So, so here we're basically the same thing, but there's one addition. First of all, the kidney, the renin, angiotensin, and then you got angiotensin 1, angiotensin 2. And what angiotensin 2 does, which is this thing here, okay, all the things we talked about, are all there to increase the blood pressure. So here we have another enzyme called ACE. And ACE stands for angiotensin converting, sorry, so hang on. No, I've got to go back a step. The angiotensin 1 is converted to angiotensin 2 through this angiotensin converting enzyme. But there is another hormone called ACE inhibitor. ACE inhibitor. It's not here. It's not on the slide. And what this is a tablet that a doctor would give you to lower your blood pressure if it's a bit too high. So an ACE inhibitor stops this working. So angiotensin 1 is not converted to angiotensin 2, and then all these don't happen, and that reduces your blood pressure. It'll happen to some degree. Yeah, I mean, you don't want to have no blood pressure. You're dead then. But this is what medical researchers do: find out what each enzyme, which hormone does what, and then find something to block it or to enhance, depending on the situation. So this is the reason I gave you the two slides here. So let's look at the diseases of the kidney. So we have nephritis, that means the nephrons have a problem. We get kidney stones, which are uric acid crystals. Now we have a tiny bit of uric acid, which is there, but depends on your diet to some degree, I think. But 4 to 8% of Australians have kidney stones, and I'm told they're quite painful. But these days they can break them ultrasonically, through sound, high intensity sound. Urinary tract infections, usually in the females, because their ureters, the urethras are much shorter than the males, and it's quite easy to get an infection in the urinary tract. Urinary incontinence, I didn't realize it was so high. 1 in 25% of Australians have some degree of incontinence. Now, this is interesting. 1 in 7 Australians over 25 have at least one clinical sign of chronic kidney disease, like reduced kidney function, the presence of protein in the urine, and blood in the urine. 1 in 7. So amazing. So what happens when your kidneys fail? But before you get, this is the transplant, I might mention that when they transplant a new kidney in, you've got to, of course, get the right markers on the kidney so the body doesn't remove it, destroy it, and you take lost the word, but anyway, it's not coming. You to stop the body rejecting it, you take a drug, and then these stay there, but they're non-operational. By the way, one of the reasons for leaving it is the adrenal glands are still working, and then the new one is they put a new port in here, and it's operating. But before they get that far, you end up on dialysis. So dialysis means that they take blood out of the body into a machine. It removes the wastes through membranes, the tubes which are made of membranes, and they work exactly the same osmotically. So they have a solvent in there which attracts the material that you want to remove like the urea, the uric acid, whatever else it might be in there that you have too much of. And then the blood, the clean blood, is returned to the from the dialyzer, right? So, and it takes some hours, I don't know how many times a week to get that. So if you have kidney disease, it's not pleasant. Before I go, there's another way of doing this is they open up, they put a sort of a port into the abdominal area. They fill it once, I don't know how often, with a solution, and all the wastes are moved inside the body, in the gut area, not in the intestines, but around the intestines, and there's a lot of blood there. And then after a period of time, that fluid is removed, and it's much easier than doing this. So quite a few people are moving towards this rather than towards the abdominal system. So this is another little clip here on dialysis. Aha, okay. So we'll stop
Get
it.
delivery.
Like a shit, oh we go, gotta eat. I get mine every day, every good chicken wings to the crib. I'm sitting.
Your doctor may recommend dialysis, a procedure that removes waste products from your blood if your kidneys are not functioning properly. Healthy kidneys filter out waste, excess fluid, and electrolytes like potassium and sodium from the blood. These waste products exit the body through the urethra in the form of urine. If you have kidney disease, your kidneys gradually lose function, leading to an accumulation of harmful wastes and excess fluid in your body, increased blood pressure, and bone or blood problems. Dialysis can also be done to treat acute kidney failure, which occurs when your kidneys stop working suddenly, or to remove drugs or poisons from your body. In one type of dialysis, called hemodialysis, your doctor will use a machine called a dialyzer to clean your blood outside your body. Some weeks or months before you begin hemodialysis, your doctor will prepare a vascular access site on your body. Your doctor will create either an arteriovenous graft or an arteriovenous fistula to access your bloodstream. To begin, he or she will numb the area with local anesthesia. To construct an arteriovenous graft, your surgeon will make a small incision in your forearm and insert a soft plastic tube called a graft to connect an artery to a vein. To create an arteriovenous fistula, your surgeon will make a small incision in your skin and connect an artery with a vein to make a larger vessel called a fistula. Once your vascular access site has healed, your doctor can start hemodialysis. To begin, your dialysis nurse will insert 2 needles into your AV fistula or graft. Once the circuit is set up, your blood will flow slowly out of your body and through the dialyzer. Inside the dialyzer, your blood will encounter filtering fibers and an absorbent cleaning solution called dialysate. The fibers will remove waste and excess fluids and electrolytes from your blood. Your cleaned blood will emerge from the dialyzer and continue through the tubing back into your body through the second needle at your access site. When your hemodialysis session is complete, the needles and tubing are removed, and you can go home. In another type of dialysis, your doctor will use the membranes lining your abdomen, called the peritoneum, to clean your blood without removing it from your body. Before you begin peritoneal dialysis, you will have an operation to implant a soft tube called a catheter. Your surgeon will insert the catheter near your belly button, creating a port through which the dialysate will enter and leave your abdomen. Each peritoneal dialysis session is called an exchange. First, you will use your catheter to fill your abdomen with dialysate for a period of time determined by your doctor. Through the peritoneal membrane, waste products and extra fluid are continuously drawn out of your blood into the dialysate. Along with the waste materials it absorbed, the dialysate will drain out of your abdomen into a disposable collection bag during your exchange.
If you wear glasses or contacts, you must see this. The Japanese Nobel Prize-winning scientist in stem cell research has discovered a natural way to reclaim the 2020.
Okay. Okay. So, having gone through the mammalian system, I thought I'd introduce you to some other systems, and and there are many different ones. I just picked 5 of these. So, contractile vacuoles, the phrydia, the renal glands, the coxals, and the Malpighian tubules. So let's go through these. They're fascinating, just the way things have. I went from the most primitive organism to the higher ones just before us. So now a contractile vacuole. So things like amoeba have a thing called a contractile vacuole, which actually collects excess water. Now, try and imagine a single cell which can move around and find its own food and remove its own waste. So it doesn't have a kidney system, so it must be able to remove the water, which is constantly flowing into it. You've got a cell in water. Okay, now I mean fresh water, not salt water. And in fresh water, being a cell, the water is hypertonic, so it's flowing in to the cell. Now, if the cell didn't have some way of removing that constant water, it would just burst. Last thing the cell wants to do. So a system of these contractile vacuoles has developed, and they had to develop, otherwise it wouldn't have existed, where it just develops and water, it attracts water into it, and it gets bigger and bigger and bigger and moves to the side of the cell and removes the water. You can have 2 or 3 of these contractile vacuoles growing simultaneously, but this happens 24/7. It can't stop. As soon as the system fails or stops for some reason, the cell bursts, and that's the end of it. So this contractile vacuole, there's a process called osmoregulation. So it's because they're regulating the osmotic pressure in the cell. And the protists, like the amoeba, pro means first. So these were the first single-cell organisms. There are plenty of them around today, but they were the first. So they fill the water and then expel. So here's a little video showing how it works, or showing that it works. We'll do another one. Here we go.
You have 10 minutes to finish your research. You rush into searching on Google, then you find a title that sounds credible. You enter the page and start reading with all your attention. By the time you read through the entire
thing, you realize the content actually has nothing to do with the title.
Amoeba are hunters. They destroy paramecia whenever they get the chance. Every part of the amoeba's membrane is lethal to the quick-moving paramecia. Amoeba's method of execution is surprisingly slow and methodical. Sometimes it stalks its prey. Sometimes it patiently waits for prey to approach. When the time is right, pseudopods extend and engulf the unsuspecting paramecium. Every embrace ends with the formation of a food vacuole. In this little chamber of horrors, the paramecium first tumbles around as if puzzled by its new circumstances. Perhaps it is similar to the many tight spots the paramecia explore during a typical day of feeding. But amoeba's membrane-bound enclosure is no place for a paramecium. A well-formed food vacuole might as well be a tomb. Amoeba greets a newly captured paramecium with a barrage of molecular scissors. Slowly, bit by bit, the paramecium is digested by teams of tiny proteins. For the amoeba, attention can now shift to other potential victims. It hunts and digests without a second thought. Of course, it never had a first thought because this single-celled killer doesn't have a brain. For the paramecium, the overriding goal is to escape, and do so quickly. Yet as protein damage accumulates, the life of the paramecium drains away. Contractile vacuoles stop pumping, cilia wave more slowly, protein construction grinds to a halt. With a final shudder, the paramecium expires. One more little life extinguished from the universe by a hungry amoeba. The paramecium is now a lifeless bundle of parts for amoeba. As nutrients are transported into the cytoplasm, the paramecium corpse shrinks to nothing, and the amoeba grows. Paramecium parts now become building materials for amoeba construction projects. Like all life forms, an amoeba utilizes information stored in DNA. It makes messenger RNA recipes for microtubules and chopper proteins. It assembles new membrane receptor proteins using amino acids from its microscopic prey. It might even reproduce by mitosis. An amoeba is powered by the combustion of sugar. Floating in its cytoplasm are hundreds of miniature power plants. Called mitochondria, they generate reliable energy to animate amoeba. Like most life forms, an amoeba exchanges oxygen for carbon dioxide. Drifting in from its watery environment, oxygen enters mitochondria and participates in the destruction of glucose. Carbon dioxide waste leaves amoeba through its membrane. An amoeba's life is the result of a complex molecular dance. Lifeless molecules collide, and often enough, the collisions change one or both of the molecules. We call it chemistry. It is the basis of life. Unfortunately for paramecia, the molecular dance that is an amoeba's life requires a steady supply of food in the form of paramecia. Paramecia death is the price paid for an amoeba's life.
Ruben,
Yes.
how long does an amoeba or the other things live for? What's the
Oh, quite a
lifespan?
while. Quite a while. Could be months.
Okay.
But they reproduce because you get one amoeba, like I've brought up again, and it just it undergoes mitosis and separates. You've got two, but it needs energy. I saw this a month or more ago when I put this together, and it mentioned contractile vacuole, and I forgot that it didn't actually go through the detail, but it showed I forgot that it didn't actually show the removal of the wastes as well, of the whatever's left over, the paramecium. Now you saw the paramecium moving around in circles. It's got thousands of little cilia along the surface, so it moves like that. So it just continues swings, whereas the amoeba just crawls along whatever surface there is there to crawl along, and it does this thing. But it looks like that the contractile vacuole, and it's a very, very efficient way of kidney. It's a healthy looking organism, and it just keeps going. Now there's earthworms, flatworms, arthropods. That means anything with a jointed leg, mollusks. They all have these excretory systems called nephridia. That's a plural of nephridium, and which is an analog of kidneys in vertebrates. So these nephridia are porous. They're long, they're thin, coiled, and they're found in all the segments except the first three. So here's a worm. And this is just an example, of course. So you've got blood going through. Here are the nephridia there. They're the ducts, okay? And the first three segments of the mouth, they don't have those. And then they move the cleaned, they move the waste into their intestines and out the other end. But it's a very early idea of the kidney. And notice that they use the same word is used, nephridia, as in nephron that we have. Okay. Then mollusks, things shellfish, things with shells like oysters, and they have single shells or double shells. They're much better developed. They use a thing called tubular. Now these ones are called just nephridia. These are called tubular nephridia. and these tubules are organized like our kidneys. They collect the liquid wastes from the coelom. Now, C-O-E-L is we pronounce it as seal, and it's a space. And we've got various seals in our brain and other parts of the body. It's just a space. And these wastes from the coelom, the space, I've got a diagram, they dump the waste into the mantle cavity, which is sort of under the shell. And they're pumped out, of the shell from there. So here's sort of a shellfish. So here's this muscular leg. The mouth has got a thing called a radula. It doesn't have teeth, but they're like grinding things like that. And they grind whatever it is, often swallow it. Now, this is their digestive system. This is their breathing system. There's a heart, a single heart, and it's basically this. There's a seal there, the coelom, and and they're not very big, and they're able to get the oxygen is exchanged with the carbon dioxide here and moved into the body of it. Down this end here of the seal is the nephridium, and this is what where the wastes are removed into this tube here, this next to it. Okay, this is the anus. So just this empty space, if you like, where the gills and the anus is. Then the third type, we're moving on in evolutionary time, called the coxal gland in arthropods, so the insects, for example. And they have a pair of excretory organs, and they consist of a sac, an end sac, where initial urine is collected, and then the tubule where secretion is and reabsorption take place, and then there's a pore called the excretory pore at the base, called the coxa, of one of the legs. So some arthropods are segmented, and and the current thinking is that each segment in the ancestors of arthropods had a pair of coxal glands. So they look like this, and then down here are the yellow things here are the coxal glands, and there's another set there. And because it's a space here, they're able to just do an exchange of wastes and so on. So they work their way through, and they have an anus. So again, it's very basic, but much more advanced than the previous ones. And then Malpighian tubules are, I mentioned insects before, but the arthropods, they're all arthropods really, they're just jointed legs. But some of them, the more ancient ones, have this system, the more modern ones have the Malpighian tubules. And they're in the abdominal cavity, and then they empty into this junction between the midgut and the hindgut. Now, in some species, this urine is acidified in the tail end of the tubule and an aqueous suspension. Now, it's not a solution, it's a suspension of uric acid. So the uric acid crystals are in this suspension so it can be removed, and then the water evaporates and you end up with a solid. And so the water and nutrients are conserved by reabsorption there, but some of them don't. So it depends, because they're all different stages of evolution. But we go into the rectum just a little bit at the end of the animal. The water and nutrients are reabsorbed, and then the solid is like fly spots or specks. So down this end here, these are the Malpighian tubules. You see them here. All right. There's the rectum, and feces and urine are here. Most of it is reabsorbed. It's useful, and the rest is pretty much just reduced, removed. Because mainly the important things that they need is reabsorbed. Okay, so this is in the mid-gut, and the rectum is at the other end. I think that's pretty much it there. Yep. Okay. So I finished a bit earlier than I expected, but it's one of the problems I had when I did this one, especially the area on blood pressure and so on. I didn't know what complexity to get into, and I hope you would follow it. I tried to simplify it as much as I could, especially the idea of giving medication for high blood pressure and the molecular, the molecules that are involved in that, because it's everyday molecules, angiotensin 1 and 2, just to maintain our blood pressure, maintain everything else around that keeps us going. But sometimes people do end up with low blood pressure, which the kidney can solve quite easily by retaining fluid, or if the high blood pressure, it has a problem, especially if it's too high. That's where the doctors start writing scripts, and that's about it. The rest, you've got the notes. You can go through the video links again, and yeah, it's still recording, so that's good. And I'll when we finish, I'll put it onto YouTube and send you the link to that. And for what it's worth, you're welcome to it. And anybody who isn't couldn't make it today, I've had one apology. I've got access to it.
Ruben,
yeah,
is everybody aware that you have a YouTube channel? Have you sent the?
Yes, every week. So every time I get I put a talk, a recording onto YouTube, I send the link.
Yes, but you can't subscribe to your channel, so you get an automatic.
I don't know. I have no idea.
Well, you can't subscribe because I've subscribed to your channel, and I will get a message when you put that YouTube up.
Oh, how do we do that?
Oh, you just got to go to YouTube and go to your channel.
Yes.
And subscribe. You just got to click on a subscription.
But how do I tell people what my channel is?
Well, it's Sue and Ruben, isn't it? Sue and Ruben.
Pat. Yeah.
Oh, is it Pat?
I don't know. Okay. I mean, I can get to see. I can see. I can go into my channel. I have to have a look. I didn't know you could do that.
Yeah. Well, everybody can go into your channel.
Yeah, but it tells me how many times my talks have been viewed.
Yes.
And so on. And so that's interesting. So I don't know who's viewing them. It doesn't tell me that. But
oh no! I've just started my own channel.
You do?
Yeah.
Oh.
Well, you got to do something with this COVID about.
So I'm just wondering then. So you tell your friends your channel address. You give them.
Yes, but whenever you go into YouTube, there's a search box, and you can search for subjects.
Okay.
And it comes up with a list of possible sites.
Right. Okay.
So if you go and look for U3A Glenira,
yes,
you should find some connection to you. Well, I don't know. It depends what.
I do call it U3A, but yeah.
Yeah.
And all right.
But
I'll have a look. I will have a look, and then send you some sort of, if I can find it. Maybe I'll Google how to do it. I didn't think I could do it. So this is all new to me.
This is all new.
Well, I learned how to put it onto YouTube. So
Yeah.
the next step is to let people know. So is there anything, does anyone have any question that I might be able to answer from the talk?
I got a comment.
Go on.
Ruben, just for everybody's information, I have dialysis 3 times a week, and that takes up each session's about 5 or 6 hours. So it's 3 times a week, and that's a quarter of my life sort of thing. And to keep it alive, anyway.
Have you tried the peritoneal idea?
I chose not to do that.
Okay.
For a variety of reasons, but I chose not to do it that way.
Do you have any idea how often that has to be done?
That's every day.
The peritoneal one?
I believe so.
Okay.
You do it at home in bed. If you've got to end up having a house modified for water and electricity and waste components and things like that.
Oh, okay. Oh, all right.
And you've got to have things in a sterile state. And having worked in a lab where sterility was paramount years and years and years ago, one of the reasons I chose not to do it.
Interesting. Okay. Well, so it's now 11:15, so officially 1.5 hours lesson done. If I continue this next year, because remember I mentioned before it would turned up a few of us, I was saying that I got an email yesterday from U3A asking me to say what I want to do, what I want to present for next year. And I can't see us having face-to-face yet next year, especially what's happening, especially if people go to engagement parties. And so I'm happy to continue. I'm writing a new one, partway through that, and hope to present that next time. The one that Peter suggested, and it's taking a lot longer than I expected to do. But anyway, so it's a brand new one. And again, with this one, I think I'll have 26 lessons once I finish this new one. If anyone's come up, Anna's given me a couple of ideas before, which I've done, but if anyone can think of anything that they're interested in, I can always expand it or add it to something else. Or my wife suggested, Pat suggested, I go through my year 11 and year 12 textbooks. Just flip through that and see if I get an idea. But I've been busy with this other thing. I have not opened it. It's all the biology books are there, but it just, I don't need the book for the information. I just need the idea. And
Ruben,
yeah,
I'd like to congratulate you on the wonderful talks that you've given. You're talking about year 11 and year 12. I did 6 years of medicine, and I can assure you that the standard that you're giving in our talks is well up to what we did in our medical course. And it's been a wonderful refresher for me, because all that kidney research was being done at Melbourne University by Professor the physiology department of Melbourne Uni under Pansy Wright and his cohorts there. And so we were very familiar with that kidney research. They had sheep, they're doing it on sheep in those days, but this is back in the 50s and 60s. But the standard that you're telling us and teaching us in these lectures has been right up to the standard that we learnt in medicine in those days. I guess these medical students these days have it a bit more difficult, but it's a very high standard talk, and congratulations to you.
Yeah, that's correct. Thank you. Thank you very much, Ruben.
Yeah, thank you. Thank you, Richard.
It's wonderful.
Yeah.
Thanks.
Okay.
Very informative and deep.
Basil's talking, but he's
muted.
Basil, press the space bar and hold it. Where is he? Oh, Basil. He has vision problems. If you see him sometimes with a magnifying glass looking at what's. Basil, press the space bar. I can't say anymore. Anyway.
Ruben, would he actually understand if you send it in the chat? Does he read through the chat link?
I think it's a bit too small for him, but
Oh, okay. All
yeah,
right.
I don't think it's worth it. No. It actually, if you look at, I've got one chat when Rick left, he thanked me, and I don't think Basil would have a chance of reading that.
Oh, okay.
Yeah, he gets, he's got all sorts of eye problems. He gets regular injections and yeah. All right. Well, in that case,
thank you.
Thank you.
I'll say fare thee well, and
Thank
all
you.
being well, I will see you in a fortnight.
No more.
Thank you very much.
Thank you. Bye-bye. Have a good
Bye.
fortnight.
Thank you. You too. You too. All the very, very best.
Can you hear me now, Ruben?
Oh yes, Basil.
Oh,
Some people have left.
I had difficulty in the beginning, and I was a little late in getting the communication going. That was a great talk, Ruben.
Thank you.
Really, you've always put an enormous amount of work into studying and getting up to date. You have to be, admired for that. Really good. And for everybody that's still on the channel, I went to Ruben's first one where there were 3 people in the first course, and Ruben did exactly the amount of work for 3 people as he did that he has been doing up to now. He doesn't stop, and they're all different.
Thank you, Basil. We'll see you in a fortnight.
We'll see you.
Bye-bye.
Bye.
Bye-bye.
All right.